morning, bom dia, buenos días. Muito obrigado, profesor Gerson, a profesora Elisa y a la organización de este maravilloso curso. It's a nice course for bringing me to Rio. Unfortunately, I have to return today to Montevideo due to issues in the university, but I'm very happy to be able to deliver this talk, and I'll be happy to interact with you during the talk. And of course, after the talk, through the email, if it's necessary, we can continue discussions, exchanging of papers, etc. So I'm very happy to be here, and I hope that you have a very good week of this rather important course for Latin America and over Latin America. So what I was requested to cover in this class was to discuss some issues related to protein oxidation and how can this relate to alterations in folding and conformation, and potentially how can this impact in disease processes. So I'm going to give an overview, and I will provide a few examples, and of course there is a lot that can be said. We can have just a whole course on this particular class. So this will be just a little bit for you to see, and of course we can continue later on. And I will talk about biochemical mechanisms of oxidation of proteins and some of the effects on protein structure and function. So the outline of the class, the first part of the class, I will take time to define the formation of oxidants and free radicals in biology, how these oxidants react with constituent amino acids, and even with prosthetic groups that lead to oxidative modifications, how then the modifications in amino acids and or in prosthetic groups can impact in protein structure and then facilitate changes in function or aggregation or fragmentation. Then the second part of all the potential oxidative modifications that can occur, I will concentrate in just one example, which is protein tyrosine nitration. This is the nitration of a tyrosine residue. Discuss some of the nitration processes in aging and disease. Then concentrate in how mitochondria constitute a central loci for nitration reactions, and then we'll provide one example of the nitration of a central protein, which is manganese superoxide dismutase, which is oxidized in vivo and in vitro and leads to profound changes in cellular homeostasis and potentially triggers cell death. So Bruce Freeman, that then was my postdoctoral mentor early on, proposed an overall scheme indicating that in biology, in cells and extracellular milieu, oxidants and free radicals are produced continuously throughout life, and also our cells and tissues are exposed to oxidants and free radicals environmentally, either by high energy irradiation, pollution, and even toxic chemicals and xenobiotics coming into our bodies are activated and lead to the formation of free radicals. So we have endogenous and exogenous sources of oxidants and free radicals throughout all our life, and this can even be enhanced under a variety of pathological conditions that increase oxidant formation. Therefore, our proteins are all the time exposed to a constant flux of oxidants and free radicals, and some of these fluxes can even increase tenfold under particular situations such as inflammation. As you can see, various organelles and areas of the cells form a variety of radicals. Of course, in each loci, the preferential radicals differ, but in general, in every single place you look for, there is oxidant formation that can impact in amino acid and prosthetic groups. And today, I will, at the very end of the talk, concentrate on oxidant formation in mitochondria and the oxidation of some mitochondrial proteins that then become biologically relevant. So, oxidants can be formed by a variety of mechanisms, and the simplest way to form oxidants is the one electron reduction of molecular oxygen leading to the formation of the superoxide radical anion. This free radical is formed continuously in our bodies and then can then dismutate to form hydrogen peroxide, which is not a free radical, it's an oxidant, and in general it's a 2-electron oxidant. And then hydrogen peroxide, in the presence of metal centers, especially transition metals, copper, manganese, iron, can evolve to a highly potent oxidizing intermediate, the hydroxyl radical, that then can get reduced to form water. Normally, mitochondria in the cytochrome oxidase reduces molecular oxygen directly all the way to water, avoiding the formation of reactive intermediates. So most of the oxygen that we consume in mitochondria goes directly from oxygen to water. But there is a percentage of electrons that leak out, or purposely some enzymes that lead to the formation of superoxide and hydrogen peroxide, and these two lead to the formation of hydroxyl radical. In general, these are called oxygen-free radicals or reactive oxygen species. However, as I said, hydrogen peroxide is not a free radical per se, but it is a strong oxidant. But what is a free radical? If I go back, this is a free radical indicated by this dot that indicates an unpaired electron, and this dot indicates an unpaired electron in the upper orbitals. A free radical is defined as a species that contains one or more unpaired electrons occupying an atomic or molecular orbital by itself. Molecular oxygen is a free radical, not very reactive, but for example, superoxide and nitric oxide are free radicals. This free radical nature can be translated to protein to form protein radicals. So if a one-electron oxidant hits a protein, then what you do is you form a protein-derived radical, which can be an amino acid-derived radical, and then continue the free radical reactions, therefore. Of course, we have a series of mechanisms to cope for the excess formation of oxidants. Superoxide dismutases eliminate superoxide very efficiently, and I will talk at the very end of the talk of an oxidative modification of one of the superoxide dismutases that compromises its function. Hydrogen peroxide is eliminated by catalase, glutathione peroxidase, and the peroxyredoxins. And then hydroxyl radicals cannot be enzymatically eliminated because they are too reactive. They are eliminated by the use of low molecular weight scavengers. So there are a series of mechanisms of detoxification, but even with these mechanisms of detoxification, there is a given amount of oxidants that still react with proteins and lead to protein oxidation. And as I said, one of the key sources of superoxide and hydrogen peroxide and peroxynitrite is mitochondria via electron leakage from some of the respiratory complexes to oxygen to form superoxide. While most of the electrons flow from NADH all the way to the cytochrome A3 to do the tetravalent reduction of oxygen to water, there is a percentage of electrons that leak out, and even dysfunctional mitochondria or mitochondria that is compromised during disease conditions increase that flux many folds. So mitochondria is a central source of oxidants. Therefore, mitochondrial proteins are typically many times oxidized. Another series of oxidants come from nitric oxide. Nitric oxide normally in our tissues is formed enzymatically by the oxidation of the guanidino group of arginine, leading to the formation of citrulline and nitric oxide. And nitric oxide in general is a signaling molecule. But it's a free radical. It has its own reactivity. And more than that, nitric oxide can evolve to secondary species, what we call nitric oxide-derived oxidants, including nitrogen dioxide radical. The reaction of nitric oxide with superoxide radicals lead to the formation of peroxynitrite, which is a potent oxidant. Nitric oxide can evolve to species with nitrosating capacity, such as nitrosonium-like, and then nitric oxide can react with radicals leading to the formation of nitroso adducts or metal centers leading to the formation of nitrosyl metal complexes. So from nitric oxide, you can go out and form a variety of secondary species that can also participate in protein oxidation, most notably nitrogen dioxide, peroxynitrite, and nitrosonium. This will lead to nitration reactions, and this will lead to nitrosation reactions. So we are talking about oxidation, nitration, nitrosation. We will define a little bit more on detail. And when it gets to nitrosation and nitration, and I'm specific on this because on the literature there is a big confusion about these two terms. And these reactions come from nitric oxide-derived intermediates. Okay? Another actor, another key player in oxidation reactions, especially during inflammation, protein oxidation during inflammatory conditions, are the heme peroxidase-dependent reactions, most notably myeloperoxidase that comes from neutrophil degranulation and eosinophil peroxidase that comes from eosinophil degranulation during inflammatory processes. And these heme peroxidases, like myeloperoxidase, liberated by activated neutrophils, normally become oxidized by hydrogen peroxide to a ferryl derivative, oxoferryl derivative, in a 2-electron oxidation process. This is what's called compound 1 of the myeloperoxidase. And then the compound 1 oxidizes chloride to form hypochlorous acid. This is bleach. And we form bleach all the time when bacteria invades our cells and inflammatory cells are activated. And bleach is a good way to oxidize and chlorinate proteins. So in an inflammatory loci, you will find chlorinated and oxidized proteins because of the bleach release upon neutrophil activation. Of course, you can form hypochlorous acid, but this enzyme can also perform 1-electron oxidation. So the compound 1 can attack nitrite coming from nitric oxide to form nitrogen dioxide. And then the compound 2, which is the ferryl species, the +4, attacks tyrosine to form tyrosyl radical or ascorbate radical, etc. So the heme peroxidases can do 2-electron oxidations to form bleach or 1-electron oxidations to form radicals, including amino acid-derived radicals, such as tyrosine-derived radicals. So if there is a lot of myeloperoxidase release, you will have oxidation chemistry in proteins, both via hypochlorous acid and via a secondary free radical initiation by amino acid oxidation. 2-electron oxidation, 1-electron oxidation. Another critical player in protein oxidation are metals. And I'm sure that with Claudio Fernandez, you will talk a lot about the copper, the role of copper in protein aggregation. There is a lot of relevance of iron, hemine, and free iron release, which participates in a variety of free radical dependent processes. I will not get into detail, but if iron is released from its normal storage sites, then iron can become redox active. It can bind to a variety of proteins and initiate protein oxidation in situ, what we call site-specific metal-directed oxidation in a variety of proteins. So, free radical processes and oxidation processes for biomolecules, including proteins, involve initiation. Then, after the initiation, you have a propagation phase where oxygen is consumed. So when you have protein oxidation, you many times have peroxides derived from proteins, what we call protein hydroperoxides, which are proteins which have bound molecular oxygen and then evolve to peroxidize amino acids. And then you have termination reactions. So, just to summarize what we have talked about, oxidant formation, and these are the oxidants that can attack amino acids and prosthetic groups. You have molecular oxygen evolving to superoxide radical, then superoxide radical dismutates to hydrogen peroxide. Hydrogen peroxide can be reduced to form hydroxyl radicals, and then hydroxyl radical can take an electron and go into water. This is a strong oxidant. Superoxide dismutases eliminate superoxide. Hydrogen peroxide is eliminated by peroxidases and catalase. And then superoxide can also react with nitric oxide. This is the crosstalk between the nitric oxide and superoxide pathways to form peroxynitrite that then liberates secondary radicals. Superoxide can also attack prosthetic groups. We will give the example of a conitase. So this is the oxidation of a redox center in a protein, not of an amino acid, but the prosthetic group. And then, as we said, hydrogen peroxide can also be substrate of myeloperoxidase to form hypochlorous acid. So this is sort of the overall scheme of oxidant formation detoxification. The biological half-lives and the reactivity of oxidants and radicals in biology are quite different. And therefore, their capacity to oxidize proteins and to diffuse distances to oxidize proteins far from the site of formation of the oxidant is very variable. Some oxidants have a really, really short half-life, 1 nanosecond, hydroxyl radical. So if you form a hydroxyl radical in a cell, it will attack a vicinal amino acid within 2-3 molecular diameters. This is it. It will not diffuse too much. However, other radicals, such as nitric oxide, can diffuse substantial amounts because of the half-life, and then peroxynitrate and hydrogen peroxide have half-lives, biological half-lives in the range of milliseconds. So that means it can diffuse 1 to 2 cell diameters. I'm saying this because one cell can be, let's say, a neutrophil can form oxidants, and these oxidants can diffuse to a bacteria, and then the proteins in the bacteria get oxidized. But you can do this with oxidants that have half-lives in the order of milliseconds. If you have half-lives in the order of microseconds, the oxidant will not be able to diffuse from one cell to the other, but rather than attack a protein which is very close to the formation site. I say this because there is a lot of, many times you attribute protein oxidation to X source, but this cannot happen just because physicochemically it cannot diffuse so much. So there is a lot of site-specific oxidation, which is metal-mediated also. This is why the presence of metals is very important for oxidation in proteins. So oxidants then can attack biomolecules, including proteins, and protein oxidation can lead mainly to two major functional effects. Well, in many cases, you oxidize a protein and it's not very relevant, either because the amount of oxidation is very small or because you are oxidizing a non-critical amino acid, so nothing happens. But in many cases, if you oxidize critical amino acids, you can have oxidative signaling or oxidative damage. And we now know that the two things occur biologically. For a long time, many investigators, including ourselves, attributed oxidation of proteins just to damage. Now we know that there is a level of oxidation that can be good for us, and this is what's called oxidative signaling. But in any case, both oxidative signaling and oxidative damage relies on the formation of oxidants that are reacting with an amino acid. This amino acid is oxidized, and this oxidized amino acid can then participate either in signaling, or this can be part of a damage process that can be propagated. Okay? For example, it's very clear now that the oxidation of peroxyredoxins is a critical signaling event in cells. So again, from Freeman and Crapo, this was just a hypothesis that has been proved through the years. There is a lot that can happen during oxidative reactions, but one is protein-protein cross-linking is mediated by oxidation reactions, proteins transition, lipid-protein cross-linking. When we are talking about protein oxidation, also we will involve other intermediates that can then form complex adducts. Let's say lipid-derived oxidation products reacting with proteins, leading to the formation of novel protein-lipid adducts, etc. So amino acid oxidation, protein-protein cross-linking, protein fragmentation, lipid-protein cross-linking, etc. And you can see much of this in conditions such as aging and neurodegeneration. You can go and you can see the protein aggregates, and these protein aggregates contain oxidized amino acid, aldehydes bound to the proteins, metals. So there is a real oxidative war that happens, and that there is a lot of chemistry that is going on in these sites of protein oxidation and aggregation. So, what then do oxidants do to amino acids and to prosthetic groups? I will not talk too much about prosthetic groups because of time. I will just show one example. But in terms of amino acid, many amino acids can become oxidized, and not all of them have the same susceptibility to oxidation. For example, cysteine is an amino acid that tends to be oxidized very easily. And in fact, is one of the central target amino acids for the signaling actions of hydrogen peroxide. So you go from cysteine to cysteine, which is the disulfide, and you can go back to cysteine. But of course, you can have over-oxidation products. We will discuss this. Methionine, you form methionine sulfoxide, and there is a lot of accumulation of methionine disulfide in aged protein. And in fact, there is a methionine sulfoxide reductase that repairs the oxidized protein. Tyrosine is another oxidized amino acid. You can form dityrosine, which is the cross-link product of two tyrosyl radicals. So a good way to do covalent cross-linking between proteins is to form dityrosine bridges. So this is a good way to covalently promote the formation of protein aggregates. Tryptophan, kynurenine, etc., and as you see, many others. So there are a lot of modifications that you can see, you can fish, and you can measure in oxidized proteins in vitro and in vivo. And you can quantitate now with the power of HPLC coupled to MS with appropriate standards. You can measure the amount in nanomoles per milligram protein of each of these modifications if you wish. So, sulfur oxidation, as we said, when you oxidize a protein, or when you look for oxidized proteins in vivo, you can go to look for carbonyls. Carbonyls come from a variety of oxidation of amino acids, and there are antibodies against carbonylated proteins that you can utilize, and carbonyls can come from the oxidation of tryptophan, the oxidation of histidine. So it's sort of a general index, carbonylation as an index of ring opening and exposure of keto groups that then you can measure immunochemically, for example. Tyrosine crosslink, tryptophan modification. We said the formation of protein hydroperoxides, especially from aliphatic amino acids, chloramines from hypochlorous acid reaction, amino acid interconversion through oxidation reactions, adducts with lipid peroxidation products such as malondialdehyde, hydroxynonenal, amino acid oxidation products that you can even measure in the urine, such as para-hydroxyphenylacetaldehyde and para-hydroxyphenylacetic acid, which are oxidized products of tyrosine, and also glycoxidation products, which is the co-oxidation of amino acids and sugars that can lead to a glycoxidation product that you see in accumulation, for example, in diabetes and in the metabolic syndrome. There is a lot of glycooxidated proteins that you can find. So this occurs in vitro and in vivo. Now the question is how much of this is relevant for the progression of a disease, or whether this is just a biomarker. This is something that needs to be studied in each particular condition. So what are the potential biochemical consequences of protein oxidation? It can lead to changes in protein activity and function. It can lead to protein aggregation by both enhanced hydrophobic interactions and covalent cross-linking. I must say, because this is a protein folding and conformation course, that protein oxidation typically exposes hydrophobic patches in proteins. So if you have a protein, you oxidize a protein. For most protein, you expose hydrophobic patches, and this facilitates hydrophobic interactions and aggregation in top of the covalent cross-linking of dityrosine. So this is a good way to initiate aggregation, is to oxidize a protein via covalent and non-covalent interactions. Of course, because you are exposing hydrophobic patches, these proteins many times becomes acceptable proteasome-dependent degradation, because the proteasome, which is the main regulator and eliminator of oxidized proteins, detects enhanced hydrophobicity in protein surface. So many times, if you mildly oxidize a protein, this is removed. This is a good thing. It's turnover. But if you over-oxidize and you aggregate, then the proteasome cannot work because the protein doesn't fit and then accumulates. So this is the dilemma. If you oxidize a little bit, many times you have enhanced removal of an oxidized protein. But if its oxidation is too strong, you aggregate and then it cannot be removed and becomes a part of an amyloid, for example, which is this.
So in the case of the protein aggregation, these reactions are downhill. I mean, they spontaneously, the cross-linking happens spontaneously?
The dityrosine cross-linking, yes. After you form the radicals, right. Then they just, and the reaction rate is pretty fast.
I see.
Of course, you must have aesthetic access, but given that you have two tyrosines which are solvent exposed, you are cross-linking spontaneously. This is just a chemical reaction. Yeah. The rate constant is about, just to say, 10 to the 6 per molar per second, so it's pretty fast. Please ask questions if there is any doubt. I should have said this. Many oxidized proteins become autoantigens, become immunogens. In fact, in the circulation of patients that undergo strong inflammatory conditions, it has been reported, just been reported, that you can find autoantibodies against oxidized proteins. Much of this may have to do in these immune diseases, that you start having antibodies against, for example, your joints. Part of this may be related to the epitopes that come from oxidized proteins that you form autoantibodies, because these are oxidized proteins. And then, if the oxidation is too strong, such as what you see during irradiation, then you can have protein fragmentation. Both during radiotherapy, for example, or a strong exposure to UV light, especially in the southern hemisphere when we don't have a good ozone layer. If you have many hours of UV light coming into your skin and your mucosas, you start having protein fragmentation. Because the UV light hits the peptide bonds, and also you form a lot of hydroxyl radical by radiolysis of water nearby, and then you can break the peptide bond. But to see protein fragmentation, you really need to have strong oxidant conditions, such as when you see to high radiation exposure. But this is seen in vivo and in vitro. So, oxidants can be 2 electron oxidants, such as hydrogen peroxide, or 1 electron oxidant, such as hydroxyl radicals. The stronger oxidants usually are the 1 electron oxidants. I'm not going to go into much detail because there are reviews on this, but then the 1 electron oxidants can attack the protein backbone and then form a protein-derived radical, or it can attack the side chains, as we said, tyrosine, cysteine, histidine, methionine. So these 2 types of modifications occur. If the oxidation tends to be mild, the oxidation tends to concentrate in the lateral chains. So what you're going to have is what we call oxidative post-translational modification. Let's say nitrotyrosine or methionine sulfoxide. But if oxidation is very strong, you may have protein-derived radicals that can lead to the formation of hydroperoxides or even breakage of the peptide bonds, protein fragmentation. Of course, as Daniel was saying, if you have protein-derived radicals and then you have another protein-derived radicals, these 2 can react with each other and do the cross-linking reaction. And in the side chains, just to show you a few examples, we will concentrate in tyrosine. Here I won't show nitrotyrosine. I will show nitrotyrosine later, but I would like to focus your attention on this. This is a dityrosine cross-link. And the dityrosine cross-link can be intramolecular, 2 tyrosines from the same molecule forming an intramolecular tyrosine cross-link. This is not the most common, but the most common is the intermolecular dityrosine cross-link that leads to protein aggregation covalently, in addition to the hydrophobic patches exposure and then facilitation of protein aggregation. Then, if there are radicals that are field formed, hydroxyl radicals, nitrogen dioxide radicals, the protein is oxidized by 1 electron and then you form protein radicals. And if you form protein radicals, the process is pretty much not very controlled enzymatically. This is, you go into a chemical process, which is not easy to control. So it's better to minimize this. But once this happens, the protein radicals are exposed to molecular oxygen, because molecular oxygen is everywhere in our bodies, right? So oxygen is good, but sometimes oxygen has a dark side. This is the price we pay because of respiring and forming good amounts of ATP in mitochondria. There is a fraction of this oxygen that gives us trouble. This is oxygen is oxidizing ourselves throughout our lives. And part of this is this reaction. Now you form protein radicals which evolve to peroxyl radicals, either peroxyl radicals in the backbone or peroxyl radicals in the side chain, depending on where the first radical was. You remember the slides, 2 slides before. So now you have the peroxyl radicals, and then there are a series of processes that can lead to side chain oxidation products, backbone fragmentation, secondary radicals, etc., etc. I will not utilize too much time of the talk, but just to tell you that once molecular oxygen reacts with a protein-derived radical, there are a series of processes that become amplified so that potentially the damage is propagated, facilitating processes such as, for example, aggregation, glycoxidation, formation of product between lipid-derived oxidized products and proteins. Just to give you an example of how oxidants can attack prosthetic groups, we discussed about the oxidation of amino acids, but proteins are not only the sum of all the amino acids, but many proteins contain prosthetic groups. And some of these prosthetic groups are what we call oxidant-sensitive prosthetic groups. One of them being a key prosthetic group, a central group, which belongs to aconitase, which is a central enzyme in energy metabolism. As you know, aconitase is one of the enzymes of the Krebs cycle. So if you inactivate aconitase, basically you stop the Krebs cycle with all the secondary consequences. This is in the mitochondria. And then the cytosolic aconitase participates in the mobilization of iron. It's totally different function. So the active site of aconitases contains a rather interesting cubane iron-sulfur cluster, 4 iron, 4 sulfur cluster, that is oxidized by oxidants such as superoxide, leading to the formation of the 3 iron, 4 sulfur cluster. So you disrupt the cluster. You break the cluster upon reaction with superoxide. And once you break that cluster, the enzyme is not anymore active. So we know that in vitro and in vivo, excess formation of radicals lead to an inactivation of aconitase. And of course, aconitase can potentially be recovered by reassembly of the cluster in the presence of reductants. But this is a rather interesting redox cycle, which can be bent towards the left, the right, or the left, depending on the redox conditions of the milieu. So it is not only that oxidants can attack amino acids. This is a clear example of how oxidants can attack and modify prosthetic groups and lead to, in this case, enzyme inactivation. In fact, I must tell you, one of the ways we have developed in the laboratory and we utilize to measure oxidant formation in vivo is to measure remaining activity of aconitase in mitochondria. Because this is an excellent index of the amounts of oxidants that are being formed. It's a perfect thermostat of what's going on in mitochondria. And this is a reaction with superoxide. We have studied this with other oxidants as well. So I'll show you a little bit about a paper that we published quite a long time ago, but brought a new oxidant at the time to the arena of oxidation, which is the reaction product of superoxide reacting with nitric oxide, leading to the formation of peroxynitrite and peroxynitrous acid. This is a merging of the superoxide and nitric oxide routes, and then peroxynitrite and peroxynitrous acid can perform 2 electron oxidations of thiols, for example, but also can evolve to secondary radicals that can initiate 1 electron oxidation. So in a way, peroxynitrite is a rather interesting oxidant because it can do a variety of reactions, and we know that it's formed under a variety of disease conditions now. It's formed in inflammatory cells, it's formed in inflamed tissue, it's formed during neurodegeneration, cardiovascular diseases. So just for comparison, let's compare the reactivity of hydrogen peroxide, which at the time was the major 2-electron oxidant that was being considered upon for protein oxidation, and peroxynitrite. So if you're looking to the oxidation of a thiol cysteine group in, for example, albumin, I must say that albumin contains 1 single thiol group. It contains 31 cysteines, but 30 of them are forming 15 disulfide bonds. So there is just 1 cysteine that is free. So if you're looking to cysteine oxidation by hydrogen peroxide, you see this type of behavior over time, and this is the reaction. The cysteine thiolate reacting with hydrogen peroxide to form an intermediate, which is the sulfenic acid plus water. And there is a pH dependency. If you do this reaction under a variety of pH conditions, if you oxidize a thiol with hydrogen peroxide at different pHs, you see that the oxidation goes faster at more alkaline pH. Why is this? Because of the dissociation of the thiol to the thiolate. If you have more thiolate, which is the deprotonated form of cysteine, the reaction goes faster. Therefore, if you move to alkaline pH, the reaction goes faster. Normally, the pKa of cysteine, the thiol group is around 8 to 8.3. This is for free cysteine. This can change a lot in proteins. So look at this shape. So the reactions of oxidants with amino acids many times is pH dependent because of the reactants involved in the reaction, such as this. This reaction constant at pH 7.4, which is about physiologically relevant, for cysteine is about here. It's about 3 to 4 per molar per second. This is a rather slow reaction in the oxidant world. It's a reaction that takes place in minutes. You see here, 300 seconds, 600 seconds. For us, this is a very, very slow reaction. If we do the same reaction, but instead of utilizing as oxidant hydrogen peroxide, which is the dismutation product of superoxide, instead of using this oxidant, we use that oxidant, that is the right side of the scheme. Let's see what happens. The reaction rate becomes much faster, about 1,000 times faster, 5,000 at pH 7.4. We have to use stop flow spectrophotometry, which is fast kinetic techniques, because the reaction is over in a period of, this is 2.5, 15 millimolar cysteine within 300 milliseconds, the reaction is over, so we cannot use a regular spectrophotometer. We need to use fast mixing, and the reaction rate is much faster. And if we look to the pH dependency profile, completely different to hydrogen peroxide. This is a bell-shaped curve, because there are two pKa's involved in the reaction. One pKa is the pKa of the peroxynitrous acid, peroxynitrite anion, peroxynitrous acid, peroxynitrite anion dissociation, which is about 6.8, and the other pKa is the thiol-thiolate. So in this case, there are two acid-base forms that participate in reaction, one dependent on the oxidant, the other one dependent on the thiol. So again, the pH makes a difference, and remember that pH changes in biology. A phagosome, the pH can go down to 5.5, for example. So these pH shifts really can influence the amount of protein oxidation. So this is a reaction, peroxynitrate with thiolate to form the sulfenic plus nitrite. As you can see, there is a perfect straight line if you plot the velocity of the reaction of peroxynitrate versus the pKa of the thiol group, but not for everything. There are a few outliers. And this is when it gets to protein structure. In some proteins, the vicinal amino acid activate the thiol group in a way that the reaction rate is enhanced many, many folds by a factor of 10 to the fourth, for example. Look at this. This is a peroxyredoxin. Peroxyredoxins are expert enzymes for the reaction and elimination of oxidants. And they contain thiol groups, the same thiol groups that all of these compounds contain. But the rate constant is 10,000 times faster because this cysteine in the active site of peroxyredoxins is activated via a series of amino acids, basic amino acids, that decrease the pKa and also lower the activation energy of the activated complex. This is a complicated issue. I just wanted to see you that proteins have ways to modulate their reactivity towards oxidants by modifying the amino acids nearby the cysteine group. So, thiols, oxidized thiols can evolve to disulfides, can evolve to sulfenic acids. Sulfenic acids are in general not very stable, but in a few proteins they are stable. Sulfenic acid can evolve to sulfinic acids, and sulfinic acid can evolve to sulfonic acids. And all of these can be measured in biology, and as we move from the thiol to the sulfonic acid, we are moving from reduction to oxidation, and in general, the more oxidized you are, the worse the protein function is. You can have reversibility until there are one protein that contains a sulfinic acid reductase that can bring back to the thiol. But in general, once you reach sulfinic and sulfonic, these modifications in vivo are irreversible. So, 2 electron oxidations, thiol radicals, 1 electron oxidations, and these are the types of modifications you can find. I should focus on this also, which is the nitrosothiol. You may have heard about nitrosation reactions and S-nitrosation reaction. Nitrosothiols, they participate in signaling reactions. So this is another way that modification of the thiol group can lead to an oxidative modification. But in this case, nitrosation is more associated to signaling than to damage. And as you can see, the scheme is very complicated. These are all products that can be formed in oxidized proteins just from thiols. Nitrosothiols, disulfides, sulfenamides, sulfenic acids, and even reactions of thiols with electrophiles. So I'm talking about protein oxidation. But what about electrophilic reactions with proteins? Because it's relatively close. Because electrophiles are formed from oxidation reactions to lipids. And then these products can then react with proteins and lead to electrophilic adducts. So the electrophiles many times are formed from the reactions of oxidants with lipids. Now you form these aldehydes that then are electrophiles that then can react with proteins. So it's an indirect reactions of oxidants with proteins. So ketones and aldehydes and nitroalkenes, which are formed in vivo, can then react with thiols, with histidine, and then form adducts. And these lipid or electrophile protein adducts can be also formed from the lipid peroxidation reaction, which is a free radical process that leads to the formation of, for example, electrophiles such as malondialdehyde, hydroxynonenal, acrolein, etc. So you can have an oxidation process, and you're oxidizing lipids, and then this lipid oxidation leads to aldehydes, and now the aldehydes can react with the proteins, and then you have electrophilic reactions. This is another way to modify proteins in an oxidative environment. Lipid protein adducts or electrophiles protein adducts. And this leads to protein structural and functional modifications. In what's called Michael addition reactions. I'm just giving the key word so that if someone is interested can follow this up. So there is a lot now being done in electrophilic signaling. It's not only redox signaling and redox damage, but a close relative of this is electrophilic signaling and electrophilic damage. So oxidants and electrophiles in biology are very close one to the other. It's not that the oxidants attack the proteins and that's it. Oxidants also attack sugars and lipids. They form electrophiles that then backfire into the protein. Did you get the idea? Okay. Actually, this is a pretty advanced concept, but I think this is the right audience to discuss this. And moreover, not only this can happen, and this is recent data from our lab, we have shown that tyrosyl radicals forming proteins can react with lipid-derived radicals and form lipid tyrosyl adducts. We have not detected this in vivo yet. This is in vitro work. It's a rather challenging task to fish this out in vivo in samples, so we are trying to develop methods. But at least biochemically, if you form a tyrosyl radical from proteins, and you have a lipid peroxyl radical coming from lipid peroxidation occurring at the same time in a cell, these radicals can react with another in a radical-radical recombination reaction and form the tyrosyl lipid adducts. And once you form the lipid tyrosyl adducts, you see the tyrosyl and lipid adducts, you form these adducts. We have seen that these adducts with time cyclicize and form a tricyclic structure. And this tricyclic structure can then evolve to secondary products. I'm not going to get into any detail, but there is a lot of rich chemistry in this. And what we think is that these reactions can facilitate also protein cross-linking. Because the tyrosine forms an adduct with a lipid, and then this lipid becomes reduced, and then the reduced tyrosyl then becomes an electrophile that reacts with a secondary protein and then facilitate protein-lipid-protein complex cross-links. There is a lot to be done on this. Just wanted to show you how the lipid and protein oxidation pathways can be intertwined. It's not just protein oxidation only. And if you get, just to look at one amino acid oxidation, there are lots of things you can evolve to. So you can see the complexity, and of course this requires a lot of bioanalytical power to define the modifications and then potentially to quantitate the modification. And then of course, does that anything to do with changes in structure and function? Because the fact that the protein is oxidized doesn't necessarily mean that this will cause any change in structure or function. So it needs to be studied case by case. So, the second part of the talk, which will be shorter than the first, will be just to focus on one oxidative modification, which is the nitration of tyrosine. And the nitration of tyrosine is the reaction of tyrosyl radical with nitrogen dioxide to form nitrotyrosine. First thing, the pKa of the OH group of the phenol of the tyrosine is 10.3. Once you nitrate, it becomes 7.3. So you gain a negative charge because then this becomes deprotonated. Oh, there is a minus sign that you might not see, but it's there, it's very thin. So you go from here, which is the predominant form at pH 7.3 of tyrosine, which is the phenol. When you nitrate, you go to the phenolate because the pKa of this group drops. So just the nitration per se is providing an extra negative charge in that particular tyrosine, in addition to having an extra moiety which increases the size by about, sorry? Yes, it's in mass is 45 Daltons, and in volume is about 40 Armstrongs to the third. So there is an extra size, an extra mass, an extra size, and an extra negative charge. So now we are here. Nitration, nitrotyrosine. Nitrotyrosine facilitates exposure of hydrophobic patches, sometimes facilitates enhanced turnover by the proteasome. But most of the times, what it is seen is that nitration, which comes together with dimerization, because you come from tyrosyl radical chemistry, facilitates aggregation. And then once it becomes aggregated, it becomes proteasome resistant. It cannot be proteolyzed anymore. It's just too big. It doesn't fit into the proteasome active site. Now you have a problem. We have a protein aggregate which is insoluble, and we cannot do much with it. So we developed an antibody against nitrotyrosine, and we tested this antibody in many different pathologies. Muchas gracias, Elisa. Muchas gracias.
Rafael?
Sí.
Yeah. Just can you come back?
Please. Yeah.
Is it possible, I imagine that it would be quite likely that instead of acting, in addition to acting on the proteins, it would act directly on the aggregates because they have some,
You mean
have a more availability to
perfect?
be attacked. And
Perfect.
of course, it's a big
Perfect. What Professor Jensen says is, what about having an aggregate first and then having the oxidative modification on top of that? And this is perfectly possible, and they may coexist. So I guess that the general question, as you say, is whether oxidation occurs before or after the initial aggregation steps, which is something that needs to be investigated in each case. It's not an easy answer. But what Professor Gerson said is perfectly right. You can have the aggregates, and over the aggregates you can have oxidation as well, which then worsens everything even more because cross links are facilitated. So thank you. The way I see the issue, but this needs to be demonstrated in more detail, and this is part of what maybe we can do together in the future, is I think that oxidation serves as a seed to facilitate the first steps of aggregation, just by exposure of hydrophobic patches. But I don't think this is extensively proved at all. It's almost a hypothesis state. But what you do see, though, what you can see in vivo is the following. You utilize the antibody against carbonylated proteins or the antibody against nitrated proteins, which is this, and you can find in the atherosclerotic plaque, in the aggregates, nitrated proteins, which is the brown immunostaining. These are nitrated motoneurons during the degeneration, and this is during lung inflammation, nitrated proteins. This is some work we did over a decade ago in human samples from the university hospital, from patients undergoing thoracic surgery. These are inflammatory cells in the lymph nodes, which are strongly stained against nitrotyrosine. There is a lot of inflammation, oxidation, nitric oxide synthesis in the inflammatory sites. But then, very interesting, and this is work done by Jose Sousa, and this was work he did during his postdoctoral work with Heri Shiropoulos in the University of Pennsylvania. They found in human necropsies coming from patients in brain, in protein aggregates, for example, in this case in the Lewy bodies, they found the coexistence of nitrated synuclein in the Lewy bodies. So these are the Lewy bodies of patients with Parkinson, and this is Lewy body disease and non-Parkinson's Lewy body disease, where they can see these protein aggregates, which are also nitrated. As Professor Sherson was saying, whether the nitration was the initiation to promote the aggregation, or whether there was first an aggregation and then it was followed by oxidation, in these particular studies, it's impossible to know. This is just an observation. So these are oxidized proteins in the brain of humans that have died having a neurodegenerative diseases. And many groups think that the oxidation process is very important to the progression of the disease. It's not just a biomarker or a collateral event. But of course, this needs to be demonstrated. However, preclinical work in animals show that if you promote by drugs, for example, oxidation processes in the brain, you facilitate synuclein aggregation, precipitation, and the apparition of Parkinson-like symptoms in animals, for example. In aging, in vascular aging, also this is a very nice paper from the group of Ulrich, published in a very, very prestigious journal of medicine. Journal of Experimental Medicine is one of the top in the area of biomedical research. He was showing enhanced peroxynitrite formation in vascular aging, and the way he was indicating that peroxynitrite was being formed was looking to the footprint of the actions of peroxynitrite, which is the formation of nitrotyrosine. And this is immunogold labeling. This is utilizing the antibody against nitrotyrosine, doing electron microscopy, and all the dots indicate nitrated proteins that you see in the aged rat, in the vessels, in the aorta, for example, of the aged rat, and you don't see this on the aortas of the young animals. This is the paper. This is very interesting. And you can see a lot of staining in the nuclei and even in mitochondria, a lot of dots, indicating that there are a lot of nitrated proteins in many different organelles. So nitration reactions are occurring in the aging vessels of these animals. through the years, different groups have collected a series of proteins that have been found to be nitrated in vivo and in vitro. And one of the questions is nitration quantitatively enough to explain changes in function? For example, if you have 100 molecules of a protein and you nitrate 1% of them, that means you are nitrating one molecule, right? So the other 99 will continue to do the regular function, so you can live with that one protein oxidized eventually. This is the loss of function theory. For the loss of function theory, you need to find an oxidized protein to a sufficient amount of the population of the protein that can explain a substantial loss of activity. That means that you need to oxidize and inactivate more than 20, 30, 40%. If you oxidize and inactivate 1, 2%, the cell, nothing will happen. The other possibility is the gain of function, meaning nitration or oxidation, even of a small amount of that population of protein, triggers something new, which can be toxic. This is what's called the gain of toxic function, which is now believed to be very important in some of the proteins. Some of these proteins become oxidized and they become very aggressive in terms of cellular metabolism. One of them, for example, is the nerve growth factor. Luis Barbeito of the Institut Pasteur in Montevideo found a few years ago that this important growth factor, if it's nitrated even 0.5%, that small percentage converts this growth factor in a strongly cell death inducing protein. So instead of being healthy, the neuronal cultures die. So it becomes pro-apoptotic. Fibrinogen. Normally, for clot formation, you need to go from fibrinogen to fibrin. If you nitrate fibrinogen, and this is seen in patients with diseases of the coagulation, nitrated fibrinogen becomes a strongly procoagulant protein, etc., etc., etc. So the gain of function is an interesting concept, meaning that even a small percentage can have a strong biological impact. So, just because of the time, I will concentrate in one last point, which is the nitration of the manganese superoxide dismutase, which is the isoform of superoxide dismutase that belongs to the mitochondria. It's an essential enzyme because the double knockouts are not viable, and it's been found to be nitrated and inactivated in vivo in a variety of disease conditions to an extent that explains the loss of function. So why mitochondria is a central focus of nitrating species? Mitochondria is a central focus of nitrating species because mitochondria, as we said, is continuously forming superoxide. And nitric oxide diffuses into the mitochondria very easily, is hydrophobic, it crosses the membranes very fast, and nitric oxide, superoxide react with each other and form peroxynitrite. And what peroxynitrite does, among other things, is to nitrate the manganese superoxide dismutase. And in a seminal paper published by Joe Beckman in 1996, they found that in human samples obtained from kidneys of patients that had transplantation, and what they call the resection of kidney is an inflammation that you have allograft resection. During the resection process of the transplanted kidney, they purified mitochondria from these kidneys, and they were able to see nitration and inactivation of manganese superoxide dismutase in these kidneys of these human patients. And at the time, they proposed that nitrating species were formed inside mitochondria, and that this could facilitate the alterations of the function, and then the part of the resection can be due to these processes. Later on, the group of Yamakura found that the inactivation of manganese SOD by peroxynitrite is caused by the exclusive nitration of tyrosine 34. And tyrosine 34 is the tyrosine which is adjacent to the active site. So this started to make sense. You are nitrating the tyrosine, which is adjacent to the active site, and you inactivate the protein. But manganese superoxide dismutase contains 9 tyrosines. And these are oxidation reactions. So how can an oxidant, such as peroxynitrite, discriminate of the 9 tyrosines, that one tyrosine, which is critical for activity? This is not an obvious question. This is a non-enzymatic process. One could expect random reactions, right? But it was exclusive in tyrosine 34. So in our laboratory, Celia Quijano explored how the metal center, the manganese center in manganese superoxide dismutase, could direct and promote the site-specific nitration at tyrosine 34. And indeed, we found that the manganese was directing the nitration to that particular tyrosine, and this was due to the metal-catalyzed nitration reaction that we discussed in this article. And more recently, in collaboration with Santiago Lamas in Spain and other groups, we found that in a series of conditions in cells, you can recapitulate manganese superoxide dismutase nitration in tyrosine 34. So you have an oxidant, such as peroxynitrite, that is being formed in mitochondria during inflammation, and this is attacking manganese superoxide dismutase that you need to eliminate oxidants. So if you kill this enzyme, your problem becomes worse, because superoxide dismutase is critical for detoxification in mitochondria. And you are nitrating that particular tyrosine. Why is this? Okay, this is a dismutation reaction. Manganese 3 reacts with superoxide to form manganese 2 and oxygen. And then manganese 2 with the second molecule of superoxide, the electron goes from here to here now, and then you release hydrogen peroxide, and the manganese 3 comes back. So this is a ping-pong reaction. First substrate, first product, and a modified active site. The modified active site reacts with the second substrate, they form the second product. And this is the structure of the manganese superoxide dismutase. In humans, the manganese superoxide dismutase is a tetramer. It's a dimer of dimers. Contains this hairpin type structure, contains 9 tyrosines in yellow. And this is the manganese center. This is where the action occurs, the catalysis. This dismutation reaction occurs is in the active site. This is tyrosine 34, 5 angstroms away. So how does peroxynitrite that is formed here discriminates and reacts only here, and then leads to inactivation? Dimer of dimers. So we are showing just a dimer for simplicity. So this is the manganese. I'm not sure if you see the yellow tyrosine here. Can you see this? Yeah, this is the OH. So the channel, the access channel of superoxide comes from us to the inside. And it has to go through that tyrosine. That tyrosine is in the channel, in the pathway towards the manganese. So the superoxide is coming from the top, is going into the channel, crosses the tyrosine, and then hits the manganese. And this distance is 5 angstroms. So we thought, okay, maybe you have a nitro group there, and the nitro group impedes the penetration of superoxide. And at the same time, peroxynitrite can react with the metal first and then nitrate site-specifically tyrosine 34. So this is the structure of the active site of the manganese superoxide dismutase. This is the manganese, 3 histidines coordinating an asparagine. This is the inner sphere, and the outer sphere, here is the tyrosine 34. And this is a hydrogen bond network that fine-tunes the catalysis. You may have seen that during catalysis you need protons. Let me show you. You need protons here. Some of these protons are exchanged through the hydrogen bond network. So this hydrogen bond network is important for the proton exchange. So inner sphere, outer sphere, the distance from here to here, 5 angstroms. Not much. So the idea is the following. Peroxynitrite anion is not so different to superoxide. It's an anion, it can accommodate into the channel. The idea is that you form peroxynitrite goes into the channel as if it were superoxide. It reacts with the manganese to form a complex that then yields nitrogen dioxide and the oxo metal complex, which then facilitates the nitration reaction. What I'm saying is that the peroxynitrite is reacting with the manganese, and that reaction facilitates the nitration of the tyrosine nearby. Is that clear? So this is what we call a metal-catalyzed nitration reaction. So of course, if peroxynitrite reacts with the metals, the nitration is in the tyrosine nearby, which is the critical tyrosine. Sí.
For example, it can be, any other metal transients can do this oxidant process.
Yes.
Just not ferrous or copper.
If you substitute, good point, if you substitute this metal for iron or for copper, the reaction will occur. If you substitute this for zinc or for cobalt that do not have redox capacity, they won't do it. So we were able to exchange metals, and we were able to show that you need a transition metal center to do this redox chemistry. So you need a redox.
Like nickel, for example.
Nickel could do it. We didn't do it, but it potentially could do it. It didn't fit in the active site, but yes, nickel could do it. So in principle, any redox active metal center could do this. So we were able to see this for Alejandra, in fact, that is in the course, has seen this for iron superoxide dismutases, and even the copper superoxide dismutases happen the same thing. But if you substitute for zinc, you don't see it. For cobalt, you don't see it. For nickel, we didn't do it, but I expect you would do it. So this is a redox site-directed reaction. Tyrosine 34, metal. This is superoxide trying to reach the active site. Here is the nitro group. Here is the nitro group. The idea is the nitro group is altering, impeding the penetration of the superoxide to the active site. But let me show you a key piece of data. This was molecular dynamics calculations we did in collaboration with Darío String from the University of Buenos Aires. Here we are looking to the diffusion, and this is the last piece of data I will show you, and this is it, okay? This is the diffusion of superoxide from the outside of the protein all the way to the manganese. Okay, we are here walking through the channel. It's clear? 8 Armstrong, 7 Armstrong, 6 Armstrong, 5 Armstrong. Here is where the tyrosine is. 4, 3, 2. At 0 would be the manganese. So superoxide is coming from the outside to the inside in the native protein. It requires some energy, which it is thermal energy, not too much, and hits the metal. This is a diffusion control reaction. The reaction rate constant is about 10 to the 9 per molar per second. Very fast. Goes in, hits the metal, gets out. If in the simulation we include this, the nitro group, superoxide is coming in, but when it's approaching the tyrosine at 5 Armstrongs, you need much more energy to push it in. You have a steric hindrance. This is considering that the nitrotyrosine contains the OH group as a phenol. But about half an hour ago we said, if you nitrate, you drop the pKa of the phenol and you convert the phenol to the phenolate. So you put a negative charge. More problems. Superoxide is a negative charge. It's entering an enzyme that has a nitro group that is bulky. It doesn't allow the superoxide to fit, but in addition, it has a negative charge. You look at the energy profile. So this is impossible. So thermodynamically and mechanistically, superoxide cannot reach the manganese anymore. Therefore, the enzyme is inactivated. So we think this is the mechanism of inactivation in vivo. So the energy barrier is large in the nitro-tyrosine 34 manganese SOD because of the steric hindrance, and the deprotonated species adds electrostatic repulsion, which is superimposed over the steric effect. In fact, you see, from here you have about 8 kilocalories per mole. But from here to here, you have about 15. So the electrostatic repulsion effect energetically is even more dramatic than the steric effect. So I believe that nitration is important not only because the nitro group and enhanced hydrophobicity, but also because of the extra negative charge. And this is a nice cartoon. We are looking to the access channel again. Manganese, this is the OH of the phenol group. And these are superoxide molecules trying to go into the channel. The channel altogether is about 10 Armstrongs, and tyrosine is about halfway, 5 Armstrongs. This is the situation on the nitrotyrosine. Plus, this OH is O minus. So this is a problem. So I think that the comparison between the two shows clearly an explanation at the molecular and atomic level why this site-specific nitration can lead to inactivation. Of course, some of these processes can be regulated by antioxidants. It's not all protein oxidation forward. There are processes that repair oxidized proteins. Glutathione is a critical repair reductant of the tyrosyl radical. So if you have cells or tissues that are rich in glutathione, you have more capacity to repair radicals back and minimize oxidation. On the other hand, if you have a depletion of glutathione and ascorbate, oxidation will move forward. So this is the end. We have summarized the formation of oxidants in biology, how it reacts with amino acid, how can they promote changes in structure, aggregation, and fragmentation. We concentrated on the process of tyrosine nitration in general, in disease and aging, how mitochondria can constitute a central logi of nitrating species, and finally we provided an example of how nitration of a very important protein such as manganese superoxide dismutase can lead to inactivation. In conclusion, one would like to interfere eventually in these processes in vivo. So how can we diminish undesired protein oxidation? How can we keep some proteins away from excess oxidation and then prolong their life, their useful life? Think about neurons, for example, where it's not so easy to replace cells. Protein oxidation, aggregation. So at the end, understanding these mechanisms can help us to create mechanisms or therapies to retard, slow down protein oxidation. We will not have time to discuss this, but there is a good amount of literature in the area. Our group is also working on this, on how to diminish protein oxidation during disease conditions to slow down the progression of disease and potentially the aging process. So, a picture of the Bay of Montevideo, y muchas gracias.
Yes. When one talks about antioxidants, right, flavonoids, tannins, etc., those are like bait to the free radicals that are going around, these that react more easily with them, or what type of mechanism is it?
Your question is very welcome. These flavonoids and polyphenols do have antioxidant properties, but they don't really act as direct antioxidants in vivo. The reason being the amount of polyphenols that you can get out of a glass of wine or a meal rich in vegetables is not large enough so that the concentration of polyphenols and tannins can really make a difference in terms of oxidant scavenging. So this was very misleading in the 90s. People were thinking that consumption of polyphenols by different ways was enhancing your antioxidant capacity because directly these polyphenols were trapping radicals. You can do this in vitro. If you put polyphenols in vitro, they do work as antioxidants, but the amounts you have in vivo after drinking wine or eating vegetables is not enough. The effect of the antioxidant effect of polyphenols and the protective effects of polyphenols, for example, in cardiovascular diseases, and this is right, this is correct, is due to a variety of processes, including indirect antioxidant effects via signaling and enhance of antioxidant defenses by redox signaling at the genome. So basically, small amounts of polyphenols activate redox-sensitive transcription factors that then go to the nuclei and promote the expression of antioxidant defense genes. So it's an indirect antioxidant mechanism. But the idea of the polyphenols directly reacting as antioxidant in vivo is incorrect, in my opinion. Okay. Thank you. But they are very good for our health, though, right? Yes.
So is it possible to obtain a good amount of antioxidants using supplementation, foods, for example, or tablets, you know,
Good
C
question.
vitamin or
question.
polyphenol supplementation.
Good question.
Enough to cause the antioxidant
direct
effect,
antioxidant
anti-aging,
effect.
and so on.
This is a very difficult question because there has been a huge amount of research in the area. I must say that some tablets of natural vitamins, such as vitamin E and vitamin C, you can take them to enhance your antioxidant capacity in vivo. For example, vitamin C and also vitamin E. The problem with the vitamin E trials, the clinical trials, is that they have found that large amount of vitamin E might be potentially deleterious because of other reasons, such as they promote hemorrhages because they have anticoagulant effects that are not related to its antioxidant capacity. So the supplementation with high amounts of natural antioxidants, as it was proposed in the 90s and early 2000, 2003, 4, 5, it's now going back. So the question is, can we enhance by external supplementation and our antioxidant capacity? The answer is yes, but not necessarily a huge supplementation will be overall good for your health, because in addition to the antioxidant effect, there are other secondary effects which are not so welcome. What it's very clear that you can have strong antioxidant effects in the skin, and in fact many creams that are being used for cosmetic or protective actions in the skin and mucosas come with vitamin E, ubiquinol, and others, because they do protect from the UV light. This is very clear. But in terms of antioxidant supplementation with high amounts, I'd say that up to 250, 500 milligrams of ascorbic acid and 200 to 4 international units of vitamin E, this is safe. You enhance your antioxidant capacity and there is no harm. And the idea is that you improve certain parameters related to oxidation. But it's not demonstrated that in the long run, high supplementation will, for these particular antioxidants, will be very good. So there are a lot of research now in synthetic antioxidants, including metal-containing compounds, mitochondrial targeted antioxidants that are, and selenium containing compounds that are being used in clinical trials for a variety of diseases, including cancer, neurodegeneration, hepatic non-alcoholic liver disease. And these trials are underway now. So this is an area of research. What is very clear, though, is that if you have a balanced diet containing primary and secondary antioxidants, the data on the long run looks like you have less cancer, less cardiovascular disease. How much of this is due to antioxidant mechanisms is not clear, but it may play a role. So we don't have a really good drug at the moment, but I guess there will be one in a few years. The mitochondrial targeted antioxidants are looking very good.
Is the nitration process a random process?
A random?
A random?
Well, yeah.
Except for the superoxide dismutase.
Yeah, it's not a random
example.
process.
It's not a random. For example, if you have nitric oxide in an environment, which is, for example, an inflammatory process, you have a lot of nitric oxide because of the macrophage.
Mm-hmm.
But this, the nitric oxide can be reduced to nitrate,
Mm-hmm.
and this nitrate can be turned in nitrogen, in dioxide nitrogen, and this nitrogen dioxide can nitrate the protein.
Mm-hmm.
Is this the process? Not just because you have to, in a certain point, to oxidate you can oxidate the nitrate, right, to form the nitrogen dioxide radical, or you can take the nitrate and reduce to nitric oxide back and do the nitration as well.
Mm-hmm.
Is this process? My question is there's a balance in what kind of process this nitration will occur.
The redox, I would say that you're right. The redox environment will define whether nitric oxide move into nitration or into nitrosation. So you have NO on one side, on the other side you have the redox environment. If you have a more oxidizing redox environment, the NO will shift towards oxidation and nitration reaction. If you have NO in a more reducing environment, let's say with amounts of CSH, with amounts of NADPH, then the NO may nitrosate, or if some nitric oxide riboxygens are formed, they will be returned back to nitric oxide, as you say, via the nitrite reduction process.
But you also need a metal center, for example, to oxidate it in these two electron steps. To reach the nitrotrides.
The metals facilitate nitration, but you have non-metal containing proteins that become heavily nitrate also. So metals facilitate nitration. But for example, one of the major nitrated proteins, if you do a Western blot, a 2D gel of a tissue under basal and inflammatory conditions, you see, even under basal conditions, many proteins nitrated, the nitroproteome. So under basal conditions, you already have nitration. Of course, if you have inflammation, this will go up, and some specific proteins will increase a lot, while others not much. Some of these, for example, one of these is HSP90, which is an important chaperone, anti-apoptotic. Once you nitrate, it becomes pro-apoptotic. That one doesn't have any metal center, but it's fairly concentrated in the cytosol.